Hi, I'm Princess Senate, conference producer at Technology Networks, and I am here to introduce our next talk, the Resistant Cancer Cell Line RCCL Collection, Drug-Adapted Cancer Cell Lines as Preclinical Resistance Models of Acquired Resistance Formation. I am pleased to welcome Martin Michaelis. Martin Michaelis received his pharmacy degree and his PhD from the Goethe University, Frankfurt, Germany. He then worked as a postdoc and later a deputy group leader in the research group of Professor Jindrik Sinatel at the Institute of Medical Virology and the Dr. Petra Jo House, a private research institute in Frankfurt. In 2011, Martin joined the School of Biosciences at the University of Kent as Professor of Molecular Medicine. A warm welcome to you, Martin. Following the presentation, we will have a Q&A session and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Martin's presentation. You can also benefit from some fantastic supporting literature which you can download for free below the video player. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. Finally, before we introduce our next session, we would like to quickly ask you a question about your virtual event format preferences to help us improve our events in the future, and a brief poll should be popping up on your screen now. The options available for selection are A, keynote speeches, B, panel discussions, C, interactive workshops, D, breakout sessions. I will give you a few seconds to respond. Thank you to all who responded. Your feedback on this call is greatly appreciated and will assist us in the production of future Technology Networks online events. With that out of the way, I will now hand over to Martin to begin his presentation.
Thank you very much. So I will talk to you the next 35 minutes or so about drug-adapted cancer cell lines. I have to admit I might be a little bit biased because this is something that I have done for more than 20 years now, adapting cancer cell lines to anti-cancer drugs. And that has resulted in what we call the resistant cancer cell line collection. I will try to convince you that we are actually doing something useful and that it is a good idea what we are doing. So the problem that we are trying to address is to improve cancer therapies. In the end, the big aim always is to help cancer patients. And and what we see is many cancer patients who are diagnosed with advanced, typically metastatic therapy, they require a systemic therapy. Yeah, the cures that we achieve are usually achieved if cancer is diagnosed early. We have a local therapy, we can have surgery, we can cut it out, we have a local radiotherapy, perhaps an adjuvant drug therapy, but basically removing the initial tumor before it has spread. If that is not the case, there are very few cancers where we have a realistic chance to cure the patient just by systemic therapy. So what you see in my little stick man here, what is a reality in most patients who are diagnosed with advanced disease is that we have a cancer patient, they're diagnosed with cancer, it's metastatic, it's not actually very good, there should be more than one tumor. Then we have a therapy, can be a chemotherapy, can be other systemic therapies. We see a response. Response can be complete remission, can be partial remission, can be stable disease, but after a while, the cancer cells adapt to the therapy and we have disease progression again. So we may have 2, 3, 4 cycles of that, but in the end, these patients come to the point where we don't know how to treat them anymore. when there is no therapy left, and we really want to know what to do with these patients afterwards. So how can we get them so that their tumors respond to another line of therapy? And in order to do so, we need to know what is effective in this tumor that has acquired resistance to the previous therapies. So from what we see in our systems, usually cancer cells that have acquired resistance to a certain therapy are still sensitive, or sometimes even particularly vulnerable to some other follow-up therapies. But we don't know at this point, at the last point, we don't know what that therapy should be. So what we will need are biomarkers that tell us whether the tumor of an individual patient responds to a certain therapy or not, or helps us to also identify therapies that would normally not be used for patients with such a disease. So where do we stand with this? So fortunately, there has been major progress with what we call liquid biopsies, all sorts of things that you can take from the blood, circulating tumor cells, circulating tumor DNA, exosomes, all sorts of things that you can take out of the blood to monitor cancer evolution. And we are getting better at that. But the main problem is we can't make effective use of all the information. We see mutations, but we don't really know what they mean. In most cases, we can't use what we get out of a liquid biopsy to directly inform the therapy, the next line therapy, the ongoing therapy. And I think we need biomarkers, as I already said, that then guide the next line therapy. And I think we need preclinical model systems for identifying these biomarkers, because only in preclinical model systems we can perform systems level analysis, where we can really look at all the cancer cells. That's not possible in a patient, because if we could get all cancer cells out of a patient, we could cure them that way. We can perform functional studies that are not able in patients. We can test multiple multiple therapies in a cancer cell population. That's not possible in a patient. We can only treat a patient once. And we can establish a large number of resistance models that is necessary to get an idea of the complexity of the process. So we want to cover the complexity of the process, at least in part. And this is why we need many models of acquired resistance. And I hope I will convince you about this part later in this session. So what we do to fill, so there are a number of large cell line projects, but there is not one cell line project that focuses on acquired drug resistance. I will also try to explain later why I think acquired drug resistance is different from intrinsic drug resistance that is normally reflected in large cell line panels. And we wanted to reduce or close the gap on acquired resistance models in cancer. So we started to adapt drug-adapted cancer cell lines. We took established cancer cell lines, or we take established cancer cell lines, we adapt them to anti-cancer drugs, to the growth in the presence of anti-cancer drugs, to the growth in the presence of clinically relevant concentrations of anti-cancer drugs. So we try to adhere to the concentrations that are clinically achievable, if possible, by continuous cancer cell exposure to stepwise increasing drug concentrations. And we try to we establish readily growing resistance models. So often when you start or when you adapt with different strategies, cancer cell lines to anti-cancer drugs, you will see that they start to survive in the presence of an increased drug concentration, but they're not really growing. They're very slow. They lose fitness. And that's probably not a good model, because non-growing cancer cells are not causing problems in patients. They have to grow. Yeah, so practically it's also very difficult to work with a model that doesn't really grow. So a final step is to make sure that our resistant cells are actually growing at a reasonable rate. And by this approach, we have established in the meantime about 2,500 cancer cell lines from 16 different cancer entities. This is based on more than 100 different human parental primary cancer cell lines, and reflects, in the meantime, resistance to more than 100 anti-cancer drugs. So the basic idea is simple, I think. So we have our sensitive cancer cell line, we have sensitive cancer cells. We add our drug, and until we have a resistant subline. So because we can analyze our cell lines by all sorts of omics methods, we can find biomarkers that indicate when these cells become resistant. I think that's a very important first step, because if you have a patient, you treat them with an anti-cancer therapy. All these therapies are toxic, because that's in the nature of cancer. The cancer cells originate from the normal cells. There is a small therapeutic window. Cancer cells have very few specific targets. Almost everything that you find in the cancer cell, you will also find in the one or the other way in some normal healthy cell. So all anti-cancer therapies are toxic. So even if we just knew the drug would stop working because the cells have become resistant, and we have a biomarker that tells us that would be helpful, because we could spare the patients a therapy that is not now only toxic but not effective anymore. Since we can test new therapies, other drugs, and different forms of therapies in the resistant cancer cell sublines, we can also identify drugs that are still effective in these resistant cancer cells, or we can even identify a particular vulnerability. So drugs that might be particularly effective in these resistant cells. And ideally, the more we learn about this, we can identify biomarkers that tell us not only when the cancer cells stop responding to a therapy, but also indicate a new effective drug treatment. And by this approach, we hope that we will be able to come up with hypotheses for next-line therapies, combination of biomarkers and treatments for next-line therapies in patients for whom we currently do not have a therapy. And I think we will need such preclinical hypothesis for clinical research in cancer therapies in the future, because in the past we did big phase 3 trials with big groups A and big groups B, but the more we learn how individual cancer as a disease is, the more obvious it is. We will probably talk about very small groups, individualized therapies. So we won't get these new treatment strategies, these new clinical hypotheses, biomarker-driven hypotheses from large clinical trials. Most important question here is, is there any evidence that our approach could work? And of course, I'm convinced, I'm probably biased, but I think there is a lot of evidence that it does. So the most important drug resistance mechanism in cancer cells arguably is the expression of drug transporters that efflux anti-cancer drugs out of cells. And the most important one, and the first one that was discovered, is ABCB1. You may have heard of that also as P-glycoprotein, PGP, multi-drug resistance protein, multi-drug resistance gene 1, MDR1. And it was discovered in 1976, actually in Chinese hamster ovarian cells adapted to colchicine. Now, that shows that even the most arbitrary model system can be used in a meaningful way, because Chinese hamster ovarian cells, they are not human, they are no cancer cells, and colchicine is not an anti-cancer drug. Nevertheless, arguably the most important resistance mechanism in cancer was adapted in this very random, arbitrary system. And also the second transporter, ABCC1, you might know this as multi-drug resistance protein 1 or MRP1, was discovered in a doxorubicin adapted lung cancer cell lines. Unfortunately, I can't remember at the moment exactly which lung cancer cell line. But also for targeted therapies, so MET amplification is a major resistance mechanism against EGFR tyrosine kinase inhibitors like gefitinib. And that was discovered in gefitinib adapted lung cancer cell lines. And that also works for classical cytotoxic anti-cancer drugs. So this is a resistance mechanism against docetaxel, a taxane, that was also found in drug-adapted, or in this case, found in drug-adapted prostate cancer cell lines. And of course, we have our own examples. So this slide shows you, this is a very simplistic experiment. We have bladder cancer patients, or patients with metastatic bladder cancer are usually treated with gemcitabine and cisplatin as a first-line therapy. So these are 6. So here we see basically 6 bladder cancer, urothelial bladder cancer cell lines that have all been adapted to gemcitabine. Here we see the same 6 cell lines adapted, or sublines of the same 6 parental cell lines adapted to cisplatin, and we just look which potential second-line therapies are still effective in these sublines. And we come up with vinflunine as the most effective one, where we only see resistance in 1 of the 12 drug-adapted bladder cancer cell lines. And this happened to be at the time, and I think it still is, the approved second-line therapy for metastatic bladder cancer. So what we see in our drug-adapted cancer cell lines often reflects the clinical situation. And this is an example that you have to memorize, because we will come back to the MDM2 inhibitors. I don't know how novel actually MDM2 inhibitors still are as an anti-cancer drug class or anti-cancer drug candidates, because they have been investigated for quite a while, and the most advanced molecules undergo phase 3 clinical trials currently. And the idea of MDM2 inhibitors is to activate P53. Now, P53 is, or TP53 is, the most commonly mutated gene in cancer. It's a very important tumor suppressor that is often inactivated by mutation. However, about 50% of the cancers have functional P53. And we know that many cytotoxic therapies, they cause DNA damage and other forms of stress, and that results in the activation of p53, and we think that is involved in the anti-cancer effects. But obviously, DNA damage is terrible. That results in all sorts of negative things and has been postulated actually also to increase resistance formation. Now clever people have come up with this idea that it might be a good idea to activate p53 directly without this stress and DNA damage and toxicity. So they developed MDM2 inhibitors. MDM2 is one of the endogenous inhibitors of p53. p53, when it is activated, it acts as a transcription factor, goes into the nucleus, induces the expression of a very large number of target genes. And some of them are endogenous inhibitors like MDM2. So MDM2 binds physically to p53, keeps it in the cytosol, ubiquitinolates it, and promotes its proteasomal degradation. Molecules like this NATLIN3 MDM2 inhibitors interfere with this interaction and can activate p53 independently of this. And there was the idea, and that provoked us to start working with such compounds, that because we don't have this DNA damage that is meant to increase resistance formation, to actually delay resistance formation. So people expected less resistance formation with MDM2 inhibitors. What we found when we first worked with them was actually resistance formation is quite quick, and it's often associated with p53 mutations. And after this long introduction, I just have to say, and that, I thought there was another slide. This has been confirmed in clinical trials, or TP53 mutations as resistance mechanisms to MDM2 inhibitors has been confirmed in cancer patients in clinical trials quite a while ago. So we could predict that based on our cell line studies. Another study, another example is that of SAMHD1 as resistance factor for nucleoside analogs. So cytarabine is a prodrug that needs to be activated as a triphosphate, and SAMHD1 is a triphosphohydroxylase that can cleaves off this triphosphate here. And by this, it is a resistance factor that can tell us that specific one, I should have said. I should have started with the disease. Acute myeloid leukemia is typically treated with cytarabine and an anthracycline, daunorubicin, or something different. Now, there's no way to predict whether patients will respond to this therapy. But we, by using obviously cytarabine adapted acute myeloid leukemia cell line, we identified SAMHD1 as a biomarker, as a resistance marker that can tell us whether people should actually be treated with cytarabine or not. And I've already explained, SAMHD1 cleaves off this triphosphate. And is involved in resistance to acute myeloid leukemia. And that is the case that has been confirmed in patients, not only by us, also by others. And so it's a clinically relevant resistance mechanism that we found. The more you look into that, and if you look into more nucleoside analogues that need to be activated by triphosphorylation, it becomes more complicated. For decitabine and acute myeloid leukemia, SAMHD1 is a resistance factor. For azacitidine, interestingly, it isn't. And then it's different for different anti-leukemic drugs in different diseases. Nelarabine is a prodrug of RAG, and we find that in acute lymphoblastic leukemia, high SAMHD1 levels actually confer resistance, whereas they don't due to the same extent an acute myeloid leukemia. So there are differences between different cancer entities here. And finally, there's a difference between intrinsic and acquired resistance. We found by using another nucleoside analog, CNDEC, we used that because it was described to be an SAMECI1 inhibitor. What we found out, it wasn't. It wasn't. But still, what we could find when we established CNDEC resistant cells is that in intrinsic resistance, SAMECI1 plays a very important role. But in acquired resistance was mediated by a loss of DCK, or that catalyzes the first phosphorylation step of CNDEC, of the activation of this nucleoside into the active triphosphate nucleotide. So there is a difference between the mechanisms and intrinsic and the acquired resistance process. And they can also be very important for the identification of specific biomarkers in the acquired resistance setting. That may not be biomarkers in the intrinsic resistance setting, but may be biomarkers in the acquired resistance setting. So we think drug-adapted cancer cell lines can be used to identify and investigate clinically relevant drug resistance mechanisms, identify potential next-line therapies in combination with biomarkers, investigate the mechanisms of action of anti-cancer drugs, and you can use them obviously as a screening tool to test novel therapies. And we think drug-adapted cancer cell lines are a model in its own right that is complementary, not better or worse, but complementary to other preclinical cancer models. So, and now I want to, the last part of my talk, I want to talk a little bit about what you can learn from not only drug-adapted cancer cell lines, but cultivating cancer cell lines in the presence of drugs that you can't learn from other systems. And the most important observation is the heterogeneity in the outcome. The repeated adaptation of the same cancer cell population, we call this, or in our case, it's a cancer cell line, to the same drug results in varying resistance phenotypes. And this is my first example. We are back with an MDM2 inhibitor, idasanutlin. It doesn't only sound similar to nutlin-3, it is a very similar compound, but is the compound that has been taken forward in phase 3 clinical trials. And we also stick with acute myeloid leukemia. Acute myeloid leukemias are usually p53 wild type, so idasanutlin is in clinical trials for the treatment of acute myeloid leukemia. So what we did is we established 45 nutlin-3 resistant sublines of 4 TP53 wild type acute myeloid leukemia cell lines. And the first thing we did is we sequenced TP53. And we found something that I was very skeptical and would have been very skeptical before we did that. And that is, we found 2 cell lines which harbor, in which the parental cell lines harbor very tiny TP53 mutant subpopulations that we just select out with a nutlin-3. Now, how do we know this? First of all sublines had the same TP53 mutation. That is statistically impossible. Secondly, with next generation sequencing, we were able to sequence very deep. And then we actually could see this small fraction, less than 1% of cells that already harbor these p53 mutant subpopulations. I have no idea how that works. I have no idea how this can be maintained when we split these cell lines all the time, but it works. So we have these subpopulations in these cell lines. In the other 2 cell lines, we have what we call de novo mutations. We see all sorts of TP53 mutations, and we also see some cells that maintain TP53 wild type. But from our experiences with other NUTLIN3 adapted cell lines, we are quite sure that all the cell lines have inactivated TP53, most of them by mutations, others by other mechanisms. So, and we have the same here for the final cell line SIGM5, de novo mutations, and some cell lines that retain or maintain wild type. Now, what strikes me is the heterogeneity in the drug response to other drugs. You to have a look at here, the very first one, the NUTLIN3 adapted MV411 subline. This is one of the MV411 is one of the cell lines in which we just selected a very small subfraction of the parental cell line. So we just accept, and still the difference in the IC50s to a drug that would be used for acute myeloid leukemia and anthracycline, like doxorubicin, is more than 10-fold. If you think that we use anti-cancer drugs at the maximum tolerated dose, we can't increase the dose twofold. So 10-fold, more than 10-fold is a massive difference. If we want to treat patients in a meaningful way, we need to know which patient is this one and which patient is that one in the future. And I don't see any other system where we could learn things like that. So if you treat the same patient repeatedly, this will have different outcomes. And we see similar thing with other drugs. So here's another example. YM155 is a survivin suppressant, at least in some cancer entities and cancer cell lines. We've done a lot of work with that. It is a survivin suppressant in our neuroblastoma cell lines. Survivin has been suggested as a drug target for neuroblastoma, and YM155 has been suggested as a drug candidate for neuroblastoma. That's why we work with this. We established a number of, this is a neuroblastoma cell line, we established 10 YM155 adapted sublines. And I just want to make one point here. On-target resistance in this case is just to develop resistance against survivin depletion, what we did with survivin as siRNA. And what you can see here, the parental cell line is very sensitive to survivin depletion, but we have a subline that is even more sensitive to survivin depletion. But we also have cell lines like that one that has established a significant resistance to survivin depletion. So what I want to show you again is the heterogeneity of this resistance formation process. And we see this in the differences in the sensitivity profiles to other drugs. And I could go on for ages now, but I don't have much time left about other examples of that. So the problem really, and we will skip this because that would take it too far for today. Many examples, I show you how heterogeneous this resistance and unpredictable this resistance formation process is. So that we won't be able to predict whether a certain patient responds to a certain drug. We will need biomarkers that tell us what is going on. And finally, a little finding, PL21 is TP53 wild-type acute myeloid leukemia cell line. But it's intrinsically resistant to NUTLIN3. So it's not sensitive to NUTLIN3. We treated it for a year in the presence of NUTLIN3 for reasons that I can't explain today. And there was no change in the sensitivity to NUTLIN3. But these cells that were treated with an ineffective concentration showed different sensitivity profiles to other drugs. And that's quite interesting. So it's easier to see here. We see here there's no NUTLIN resistance, no development of NUTLIN resistance in these cells. But we see strong differences for cytarabine, doxorubicin, and gemcitabine. So this tells us basically that even if we have a first-line therapy and it doesn't have any effect, it may affect the outcome of the next line therapy. So it is very important for us to understand all these processes, and that is just, I know that's tough. When we just cultivate the cells for a year, this doesn't happen. Yeah, it has something to do with the presence of NUTLIN3. That's very important to say. So, wrapping up what I've just said. The mechanisms underlying acquired resistance formation cannot be predictive. Even non-effective therapies can affect the outcome of next-line therapies. And we all learned this with cell lines. And this is, and many of these observations that you can't make in any other system because you can't have the number of cells. And interestingly, the last TRACER-X study came to similar conclusions when it comes to the evolution of non-small cell lung cancer cells. Biomarkers are needed to guide anti-cancer therapies in response to the therapy-induced cancer cell evolution. And we need to find these biomarkers. And we think that drug-adapted cancer cell lines can be one of the tools that helps us to identify these biomarkers. Because we can do all these investigations that I've just shown where we adapt the same cancer cell line to the same drug again and again and again. And you can't do anything like this with patients who can only be treated once. So that's basically the problem. We will have cancer cells in patients and we will treat them with the same drug and the outcomes will be different. And we will have to understand this difference here and be able to have biomarkers that show us which cell we have and what is the actual next line therapy. So I think a broader message is we have to embrace the heterogeneity in cancer. We have to complement the reductionistic approaches that we have used traditionally with studies that actually investigate the complexity and variability in cancer and in response to therapies. And we feel that the drug-adapted cancer cell lines and our RCCL collection are a preclinical research tool in its own rights that is complementary to other approaches and that can help us to improve anti-cancer therapies. And of course, I'm not on my own. Many more people actually have been involved in the data that I've shown. The people I've mentioned here are mostly focused on the last bits of research, the newer bits of research that we have performed on the heterogeneity in drug-adapted cancer cell lines and the impact of non-effective drug concentrations. And with this, I'm at the end of my presentation and I'm available for questions. I will do my very best to answer all questions as good as I can.